I'm Michael Breakspear from the University of Newcastle, and I'm speaking in the educational course on time-varying functional connectivity. I'd like to thank Raphael and Vince for organising the session and the program committee for selecting our session. I'll be speaking about neural models of dynamic functional connectivity, linking scales and data modalities. So for a bit of a segue, just going to talk about weather forecasting. This puts what we're doing in neuroscience in a bit of context. So say we wake up in the morning and look out the window and see the weather. Some clouds, some blue sky, and we think, well, what should I do later in the day based on these observations? Should I pack my beach umbrella and go to the beach? Or should I pack a rain umbrella in preparation for rain? So what do? We go to the Weather Channel. Now, what does the Weather Channel do? Well, here is the world's best weather channel. It's called windy.com. It's a lovely visualization of several large supercomputers running simulations of the world's weather, and I've centered on Australia here. What you can see is that you can click between different observations. So I'm a keen surfer. Here I've clicked on waves. Here I've clicked on wind. And this is a cross-section of what is being observed and modeled in the Southern Hemisphere today. Now, how does this beautiful simulation work? Here's the simulation. It visualizes, as I said, massive supercomputers running simulations of the weather, and it does this in tiny little cubes of the atmosphere, tessellating the world's surface and then building up to the outer edges of the atmosphere. Millions of coupled little cubes. And in each cube, the computers simulate the solutions to something called the Navier-Stokes equation. Doesn't matter what the form of it is, it's a set of equations that describes the movement of gas or fluid in the presence of warming and pressure and viscosity and other things pertinent to the weather. And this is done over the land and then in subsequent towers up into the top of the atmosphere. And of course, to make this work, it needs to be integrated with multimodal weather measurements, including surface and atmospheric wind, temperature, atmospheric pressure, all the things that go into the Navier-Stokes equation. Here's a forward simulation of that model. So you can see along the bottom of the bar here, predicting weather as we go forward. And in Australia, you can see a cold front now passing over southern Tasmania. This is done by forecasting forwards, taking measurements, measuring the error, and then updating the parameters and the states of the model. This is a process of core data assimilation, and it's done every day all around the world, and that's where our weather predictions come from. It can also be used to predict storms, and with certain simulations, it can then be used to forecast long-term weather and even climate change. So what I'm going to do in the remainder of this talk is convince you that, well, I hope I've convinced you so far that this way of modelling and predicting the weather works. And now I'm going to try and convince you that we can and are doing the same thing with the brain, modelling, forecasting, measuring the brain, and then updating our models. Of course, we can't use the Navier-Stokes equation, and this is the key issue here. We need to develop models of brain activity. So just as the Navier-Stokes equation modelled wind and pressure and temperature, our neural models need to model firing rates, membrane potentials, membrane conductances, But otherwise, the nature of the models and the objective of the modeling enterprise is the same as it is with the weather. Now, it's not new to want to model brains. And in fact, in the 1950s, Hodgkin and Huxley developed a very sophisticated model of the single cell called the Hodgkin-Huxley model. It was derived from measurements of the squid giant axon, and it works so well they won the Nobel Prize for it. Here are the equations. The complicated details aren't important. It models the flow of ions across the cell membrane. So that gives rise to a current, which you can see here. Whoops, where's my model? Well, you can't see. C to V to T. That's the transmembrane current through sodium and potassium and leaky channels. And those channels flip open and closed according to the membrane potential. And using these equations, we're able to predict a whole type of different neural activity: spikes, bursts, fast spiking, erratic spiking. And I think it's a wonderful success in computational neuroscience. But what challenge do we face in our field is that individual neurons are buried within circuits of thousands of neurons, which are then immersed in functional subsystems and large-scale brain networks. So it's not really enough to model a single neuron. And even a single neuron model has got 7 different variables, and it's impossible to scale that up to something approaching the many billions of neurons in the brain. Individual spikes are too weak on their own to influence behavior. And if their precise timing was crucial, then you can imagine that the way they would code information would be so high dimensional, and it would be very, very fragile in the presence of noise. So if my brain state or my cognitive state depended on the exact state of all the neurons, and some of them were perturbed, then my cognitions would change likewise. And that's not a very stable way for the brain to operate. So it seems increasingly likely that brain function is based on population-based codes of ensemble activity, not individual spikes. So that's what I'll talk about now. Before I go on, I just point out this is not different from what is done with the weather. Forecasting the weather doesn't involve forecasting the movement of individual air molecules, oxygen and carbon dioxide molecules. It's an ensemble approach, which is captured by the Navier-Stokes equation. We're going to do exactly the same with the brain. We're not going to model individual neurons, but we're going to model ensembles of neurons. And moreover, since we're at OHBM and we're measuring the brain with fMRI, EEG and MEG, and other modalities, these population models are nicely matched to the apertures of fMRI and EEG and MEG. So here's our local neural population. If I just model, say, 1,000 neurons, then I can do this quite easily, even on a laptop with a few simplifications. And I can model thousands of spiking neurons. And what you see here is called a raster plot with the states of each neuron when they fire indicated by a dot, and then time indicated on the x-axis. If I take a cross-section of the states, you can see that they have a probability distribution. I've taken one on the left early in my simulation and one on the right later in the simulation. So the so-called mean field approximation involves taking that distribution and approximating it with a normal distribution or another simple probability distribution, and then getting rid of all the states and just modeling the mean and the variance, or the value of the states and the precision of the states. Then I have an equation to propagate those forward to predict the mean and the variance, and hopefully there's a good prediction match between the prediction and the actual simulated states. And if there is, within error, then I've got a good model of those neurons. This is exactly the same approach that's used very widely in physics, magnets, supercomputers, liquid crystal displays, curing behavior of humans, and swarming behavior of insects. So how do we do this? We take our complicated, beautiful neural histologically defined column. We abstract that down to spiking neurons with dendritic and other compartments. We abstract that further to simple spiking neurons, which we can simulate. I've just shown you those. And then we use this so-called mean field approximation just to model the mean and the variance. And in most of our models, just the mean. We keep the variance fixed. This is the mean field or dimension reduction, of which there's several approaches of various complexities. So now we've got our mean field so-called evolution equation. And the F here on the right-hand side, which brings in all the physiology, in math, that defines a vector field. A little arrow at a particular state, X and Y, could be membrane potential and firing rate. And because it's an equation defined over the whole manifold, we get a whole bunch of arrows, which we can then join together with smooth orbits, and these then collect onto so-called attractors. The simple attractor, like a limit cycle, predicts periodic behavior. We can have bursty sort of attractors, and we can have so-called chaotic attractors. These are fully deterministic, but they're aperiodic, and chaos is a lovely area of maths. I won't go into it today. Now here's our neural mass model. So this is what we've done. There's several of these. This is a so-called conductance-based one. Put it into Python or MATLAB or another simulation engine. And here's the attractor for this column of neurons. So this is a bursting state. I can change some of the parameters, and I go from bursting through to a chaotic oscillation on a so-called chaotic attractor. So that's a model for one small population of neurons. Highly abstract pyramidal cells and local inhibitory cells, no layers, nothing else. To model something approaching the brain, I now place one of these models in each parcel of the brain. So we need a parcellation, and that's covered in other talks. Here they go. I couple them together so they influence each other through the white matter, and I add terms so that the states of one influence the states of others. And I put simulations at each one and then couple them together. How do I couple them together? I use diffusion tractography to derive an estimate of whole brain connectivity. This is structural connectivity. So this is a measure of how these regions are connected to all other regions. I'm sure most of you have heard of the connectome. So I'm taking neural mass models and I'm coupling them together through the connectome, and then I'm going to simulate them forward in time. So what happens when we simulate these whole brain models? Well, almost inevitably, after a few seconds, the states, these parcels, evolve towards these attractive large-scale whole brain patterns. Here we see rotating waves from a different set of states. Here we see something called breathers. And from different states, initial states, we see another type of wave pattern called a traveling wave. These waves aren't just fictitious entities of numerical simulations. Waves have been observed and recorded in many different modalities of neuroscience data, including traveling waves of perception in humans during binocular rivalry, and multi-action units, local field potentials of non-human primates. Here's some recent recordings from surface electrode recordings in the human from Terry Sanowski's group, showing traveling waves of activity in the 10 Hertz band over the temporal and inferior frontal cortex. And here's a simulation of our model showing almost precisely the same oscillations, types of patterns, and to first order even wavefront velocities. Dynamics spontaneously cycle through these different patterns. You don't have to start. You can just watch them. They'll start in one pattern. Here's a traveling wave. And after a period of time, the pattern changes. And now it's gone to these two symmetrical rotating waves. So we can quantify that with something called an order parameter. James Roberts developed this. And on the x-axis here, you see time. And on the y-axis, you see a complicated cross-correlation function. But it captures a fingerprint of each of these unique states. When the entropy of these states goes up or the order goes down, we can capture transitions between the states. So that's the states. How do we observe these? Now, as I said before, in the case of weather, we had weather stations. And in the case of the brain, we do a hemodynamic convolution with the bold signal, and we can predict here dynamic functional connectivity that the other speakers in this session have spoken to. And this gives a temporal pattern of different changing functional connectivity matrices, which you can characterize with their graph metrics. We in fact did this. We simulated a model and we compared it with resting state functional connectivity data from the Human Connectome project to show in first order there's a match between our large-scale brain model and our observed whole brain functional connectivity matrices. We can go beyond just looking at the patterns of the waves. Here's 4 snapshots of our wave states. These define 4 vector fields, which we can link, as I showed you before, with spatio-temporal orbits. So here's a full simulation of our brain model with the waves, the accompanying vector field, and the accompanying orbits and attractors. And what happens is when there's a transition from one wave pattern to another, the attractors and the nodes and the saddles bump into each other, and then they reorganize into another pattern. And of course, we're far from understanding this. But we can use these streamlines to better understand and capture the jumps between the different whole brain states. Of course, we can change the parameters and we can get waves, we can get clustered states, we can get a combination of clusters and lurching waves. And I won't go into the detail here, but according to the different parameters of the model, we can have this whole repertoire of different dynamical states, random states, traveling waves, whole brain synchronous states, which would be grossly pathological, lurking waves and everything. Now, there's 2 packages to do these simulations. The virtual brain, which is a large collaborative effort led by Randy McIntosh, Petra Ritter, and Victor Yersa. That's a Python-based platform, which you can access via the Virtual Brain website. And a simpler toolbox that I've done with some help of a colleague, Stuart Heitman, called the Brain Dynamics Toolbox. That's MATLAB-based. You can Google either. This is a snapshot of what the Brain Dynamic Toolbox looks like. It comes shipped with a whole lot of inbuilt models. You can the time portraits, the phase portraits, and you can do some simple analyses of these patterns. It's fairly simple. You can get the code to run in just a couple of lines. There's an accompanying handbook, and it's, of course, open access, although this does run on MATLAB. If you prefer Python, then I recommend the virtual brain. I just want to end by touching on something that we're doing at the moment, which will come up in the other talks in this session, and that is moving beyond purely brain models. If we look at a human, and this is a friend of mine, Roland Girk, being filmed and sped up, what do we see? Emotion, respiration, head motion, physiology in action. How do we do this in our brain models? Well, what we do is abstract Roland to coupling between brain and heart. We then write down equations for the brain, which I've shown you. We can write down similar equations for heart activity, and then we can couple these together. And we can have observations from both of these as well. So just to conclude, I started with an introduction to large-scale simulations with the weather and the underlying Navier-Stokes equation and the notion of modeling and observing iteratively. And then I walked through pretty quickly similar models of the brain, neural field models, neural mass models, and showed how these could be integrated with different observations from functional neuroimaging data and behavior. I'd like to end by acknowledging the Systems Neuroscience Group. There's one in Brisbane, and now there's a new one in Newcastle where I've moved. Particularly James Roberts, Leo Goyol, Luca Cocchi, and Stuart Heitman, my funders. And I'd like again to thank the organisers of this educational section. Thank you.
So, hello, my name is Joana Cabral, and I'm going to talk about wandering around functionally relevant network states. So briefly, today I'm going to talk a bit about dynamical systems theory, which is a field from physics that I believe is crucial to understand a little bit more about the complexity of the brain. Then I will briefly talk about the difference between synchrony and phase locking, what is understood by phase locking. And then I will show results we have been finding and how to apply this technique to detect this whole brain dynamic functional connectivity and detect meaningful patterns from the bold signal. So, systems of interacting dynamical units, be they atoms, molecules, cells, or life forms in nature, in the cosmos, can exhibit collective behavior that cannot be easily understood by looking at a single unit alone. So you can investigate the single unit, characterize its dynamics, but then the whole, the ensemble, exhibits a complex collective dynamics that needs to be understood. So the theory of dynamical systems serves to search for the fundamental rules through equations, mathematical equations that describe processes from physics that govern these complex collective dynamics emerging at the system level. Now, when this whole, this collective ensemble, becomes more meaningful than the aggregate of its parts, it is referred to as a complex adaptive system. As we can see in this cartoon picture, a group of fish can group together in order to increase its chances of survival. So there are many processes in nature that result from this sophisticated coordination of interacting dynamical units. They can be different or identical, but where the ensemble engages in a non-trivial but functionally meaningful collective behavior. So these complex dynamical systems can exhibit multi-stability. And this multi-stability is important because it allows the system to display what is called the butterfly effect. You probably have heard about it. So the idea is that the single flapping of a butterfly wings can have dramatic impacts in the weather of the whole world. And this is interesting in the brain because we have the smallest units are neurons, and then we have the whole brain, which is a system composed of billions of interacting neurons. And we want it to be in this critical regime where the minimal change in one of its units can have dramatic consequences in the behavior of the whole system. So the most famous example of this is this pattern that is represented here. So this is the Lorentz attractor, which is a system that has two possible attractors, let's say, where the system can switch between the two, and a small change can make it transition to another state. But this is only by stability. And you can have really, they are really called strange attractors, where multiple small, weakly stable attractor states can be engaged. And this will be, I will talk about this later, but this is relevant to understand the following of, to follow the presentation, the rationale of the presentation. So how do we apply this to the brain? Why? So the idea is that the brain is a very complex network structure, perhaps one of the most complex structures in nature, and it is composed by 100 billion neurons that are connected by trillions of synapses. And this system, this structure, reflects into function by generating an infinity of activity patterns that coordinate behavior. So it's important to understand what are these patterns and how they are formed. So in the last 10, 20 years, we've seen a lot of reports showing that the brain, so the brain activity detected with the fMRI has shown the emergence of different functional subsystems. They are called functional networks that are even present during rest, so these so-called resting state networks, that show the particular patterns of activity that have been linked to brain function or to behavior. So from a physics perspective, this empirical evidence points to the existence of an adaptive complex network dynamics in brain activity. So now, enough of the theory of dynamical systems. I will come back to that later. And I just wanted to make a small parenthesis because I think it's important, and there is usually some confusion on this, and it's crucial to understand dynamic functional connectivity. So we can talk about different measures of functional connectivity. So we can talk about coactivation, that is when the bold signal in two different brain areas, here represented in blue and red, simultaneously increases and decreases, so following the hemodynamic response function shape. So this would be coactivation. Then when we have the correlation below, we can look at larger intervals of time and see how these fluctuations in bold activity are correlated or connected. But these are all talking about fluctuations. And recently there have been some papers and studies that have shown that maybe in the bold signal we can detect more than just fluctuations, but really oscillations in the bold signal. And when we have oscillations, we can talk about synchrony. So it's not just a coactivation at an instant of time, but when the signals oscillate consistently. And we can have synchrony, or we can have phase locking. So when two oscillations, when two areas are connected, because we see that there is a link in their phase relationship, but they are not necessarily in phase, right? So an example of this is a very caricatural example, but it shows that full synchrony is only one possible mode of operation of dynamical systems displaying oscillatory behavior. And there can be an infinity of phase-locked oscillatory modes. And the different phase-locking solutions can have different frequencies and, importantly, activate or reveal different parts of the system together. So one of the main hypotheses of my work recently has been to understand if the brain functional networks can be the expression of phase-locking modes in the bold signal. So how do we look at the phase of the bold signal? So when we record, imagine this is in the real axis, so over time, this would be the bold signal in one voxel or in one brain area, so averaged across a number of neighboring voxels. And we have a signal. So, but we don't have the phase. So if we just look at signal at a single TR, for example, if it's at 0, we don't know if it's going down or if it's going up. So one of the things that is used is the Hilbert transform that transforms this signal into an analytic signal that can have an associated phase. So I made a small video to illustrate this. So we imagine another dimension, and then the phase is represented here as arrows. So this is at each instant of time, we can capture the phase. So you see over time. So now if we collapse the time, we have the phase portrait of the system. So in each brain area, or each node, we can capture, using this Hilbert transform, the bold phase, the signal phase in each brain area. And in this case, we are neglecting the amplitude dynamics, so just the phase, but the Hilbert also gives the phase. And in a way, the real signal is represented by the cosine of this phase, and the imaginary part is the sine of this phase. But I will try to avoid going into deep mathematical concepts. So overall, if we look at one TR, we can see the bold signal phase in each brain area at each single time point. So here, for example, it's in 90 brain areas derived from the AAL template. And I represent each arrow is placed at the center of gravity of each brain area, and it represents the analytic phase. But this is multi-dimensional, so it's a complex vector. And how can we characterize the bold phase relationships at the whole brain level? So this is something I've been working on. And in 2017, we published this method that is called LIDA, from Leading Eigenvector Dynamics Analysis. And basically, in simple terms, what it means is that we project the bold signal phases in each brain area into the leading phase eigenvector. So this is the vector, imagine another arrow, where all the phases maximally project, as represented by the dashed line in the phase portrait over there. So this is all the phases in all the 90 brain areas, and then they are colored in red if they project in one direction of the eigenvector, and in blue if they project in the other direction. And we use a convention where the blue is always the largest community, so the largest direction, and red is the smallest community. I will come back to this later as well. So this vector that we see on the right side, this eigenvector has n elements, so the number of areas or voxels that we are considered, where each element represents the projection of the phase in each brain area into that leading eigenvector. So the ones that are perpendicular are close to 0, and the ones that are really aligned are close to 1, if they are normalized. And here, as I explained, the brain areas can be separated into subsystems, here in red and blue, according to the direction of this projection into V1. So in a way, here is another TR. So if we look at the direction of the bold signal phases, we see, for example, that in blue, they are all pointing in one direction, let's say, and then in red, we see some brain areas whose bold signal is shifted from this main direction. And this leads to a phase coherence pattern so the matrix of interactions at the instantaneous level, this is the cosine of the phase difference, specific patterns of activity. So then if we take the areas that are shifted from the main bold phase orientation and we plot it as a network, as we represent here, we can see that this network can have a specific, it seems a functionally relevant pattern, let's say. So what we do to look for these patterns is to compute this leading eigenvector at each TR from all fMRI scans. So we compute this eigenvector. And then we can see the dynamics over time. So here, for example, we see the instantaneous bold phase coherence matrix on the left, which is a matrix with n times n dimensions. And then on the right, using only the leading eigenvector, and here again plotting links between the areas that are shifted from the main bold phase orientation, we see the formation of subsystems of networks that appear recurrent, appear sometimes meaningful, but it's important to characterize them even further. So how we proceed, so if we have the bold signal in the top panel, we see the bold signal in these 90 brain areas unfiltered. In this case, we didn't filter the data. And then, as you can see in panel C, we see that this the bold phase portrait is changing over time. And especially, we see a rotation of the referential eigenvector. And by coloring the brain areas that are shifted, as you see in panel D, we can see the subsystems or the brain areas that are being activated over time. Activated or shifted. So it's unclear if indeed they are activated. So the idea is to find out, are there rules governing these bold phase locking patterns? Are they meaningful? So we took resting state data from 99 unrelated healthy participants from the Human Connectome project, which is an open source database. And what was special about this data set is that each participant was scanned twice on the same day. So we have a validation data. So here we use just the data from the first session, and we projected, so each dot in the left plot is a cloud of observations. So this is how it's called. And it is, each dot represents the brain at 1 TR. So the bold phase locking pattern detected at each TR. And then we use a k-means partition to divide this system into clusters and to identify if we can divide the system or characterize this system. Is it only one pattern? Is it bistability? Do we have multiple patterns that are co-occurring over time? How do they, so that's what we will characterize in the next slides. But importantly, when we apply a k-means partition, it returns the cluster centroid. So it divides the whole cloud into small clouds, and then we have the center of gravity of each cloud, of each cluster that is represented by a pattern, let's say. It is the central pattern in that cluster. So we see that we have different spatial configurations when we look at these brains, at these centroids, but the idea is to see, are these network patterns meaningful? And again, the story of the fish. So the idea is, even if we detect these patterns, do they have any evolutionary advantage? Why do they form? Can we relate it to the literature that exists? So we partition the cloud into K clusters, and we varied K between 2 to 20. So we first, as we see here in this plot, we first, when we cluster into just two clusters, we found one state where all brain areas are going in the same direction. So the brain here is represented as transparent, and we color only the areas that are shifted from the main signal or bold signal orientation. So we find that the second cluster, the second centroid, let's say, overlaps. Here we compare the different centroids with reference functional brain subsystems that have been reported in the literature. And from this, we use the intrinsic connectivity networks from Liegeois et al. 2011, which are here represented in panel B. So this is the visual, the somatomotor, dorsal attention, ventral attention, the limbic network, the frontoparietal, and the default mode network. And we find that as we divide into different clusters, so into 5, 6, 7, 8, 9, 10, we recognize these functionally relevant states. They have a good overlap when we color the brain areas that are shifted from the main bold signal orientation. So this gives us an intuition that there is some functional relevance. Also, to evaluate what is the best number of states into which we can divide the brain, We applied the silhouette criterion. There are different criteria to evaluate cluster performance. And it tells us that between 3 and 7, we have a good, a better, let's say, explanation of the data than if we increase the number of states. But still, I would like to point out that when we have higher number of states, for example, here we see that from k equals 7, there is a brain subsystem that appears colored in black because it is not overlapping with any functional brain system from Yale, which is composed by subcortical structures. For the following of our analysis, and since we are not interested here in specific brain configurations that relate to a specific condition, but mostly to characterize whole brain activity from this dynamical systems perspective, we took the solution with k equal 5. So here we found that we have a state which is a global state. Everything goes, the whole signal goes in one direction in those TRs. Another that overlaps with the default mode network, the second most prominent one. Then we have the frontoparietal network, which is the one in yellow. Then we have the visual network and the ventral attention network. And the values here report the overlap with reference brain subsystems. So as we see, the correlations are really strong, the p-value is really low. And so we can say that we have functionally meaningful patterns. So taking this solution with 5 states, here I represent again the arrows in cortex. And so as we see in the first one, the Bolt signals are all going in the same direction. In the state number 2, the Bolt signals in those brain areas that are colored in red are shifted, are going in the opposite direction. And in Bolt state number 3, Bolt state number 4, Bolt state number 5, the same. So below at the bottom row, we see the overlap with the reference functional networks. And as we see, this one has a very good overlap only with the default mode network. The second one has a very good overlap only with the frontoparietal network. The fourth state has the second, third, fourth has a good overlap with the visual. And then the fifth one, we find that it has overlap with both the somatomotor and the ventral attention network. But this is due as well to the reduced number of clusters that we chose, because we are comparing with 7 subsystems. So if we increase, we would also find the limbic network, which may also be relevant. So this is how the phase locking matrices, for the people who are used to look at matrices over time. So this would be the patterns of in matrix format that we would observe when we detect these states. But these states, we want to know how long do they last, because they are rarely detected in, these networks are detected normally in Bolt signal, but we don't know how long they last. And here, since we have unfiltered data with a good TR, so 0.78 seconds, we characterized their time. So one of the questions is, do these patterns occur in all subjects? Do we have consistency? And also, do they reoccur in the second same day session? And if they occur with the same probability between the patients. So we will look for individual fingerprints. So some of the measures that we take from dynamical systems theory, one of them is the fractional occupancy. And this is the equation, but I think it's more clear if we simply understand it as the proportion of time points assigned to states alpha, to any state alpha, in each single session S. So here, for example, is to count how many TRs this blue state or this specific pattern was active, and divided by the whole. Another measure that we take is the dwell time. So how long, when we are in a given state, how long does it last? Does it sustain? So it's basically the average number of consecutive time points assigned to state alpha in each session S. So how wide these blue bars are, let's say, on average in each subject. And so here below, in the two bottom plots, let's start with the fractional occupancy. So each point here represents a subject, and the X and Y axis represent the two different sessions of each subject. So we see that there is quite a So it follows the diagonal, especially for the state one. The default mode network is the second most probable of occurrence. Sometimes some subjects show up to 50% of occurrence of the default mode, whereas the frontoparietal, the visual, and the dorsal attention, they are less probable of occurring. And especially when we look at the dwell times, we see that the dwell times are really close to the TR. So they are very short-lived states when we unfilter the data. If we filter, they may appear to last longer. But with unfiltered bold signal, we find that they appear to last quite shortly. And then we want to understand, like, what is the dynamics of these states, how are the transitions? And for this, in systems of multi-stability, we use the so-called Markov chains. So basically, what we will calculate is the probability of being in state alpha switching to state beta. And better than equations is to illustrate this. So doing the Markov chain transition graph, what we find, so our matrix, what we find is that, for example, in the matrix format, we see that the probability of being in state one and remaining in state one is around 77%. Yes. So here we represent this by an arrow saying that when you are in this state, you are more able to remain in this state, more probable to remain in this state. Then from time to time, for example, here with 8% probability, it can switch to the default mode network. So this is the average across the 100 subjects. Here is not at the individual level. And when it's in the default mode network, there's around 50% chance of remaining there and then returning or switching to other states, but with very low probabilities. When you are in state two, switching to other states is very low, but switching back to state one, either you are in state two, state three, state four, state five, these arrows here are thicker, so you always have more probability of switching back to state one, which is the global mode. So we can consider that this global mode is what we call a main attractor in this whole brain dynamical system. And the different states that appear that are short-lived and there is weak probability of switching into them, but they are functionally relevant, are termed ghost attractors. And here comes the name of our paper. So it's called Ghost Attractors in Spontaneous Brain Activity. It was published now in April, and it's a work by Jakub Vorisek in collaboration with Gustavo Deco, Bruno Cesac, and Morten Kringelbach. So I invite you to take a look at that. So one of the things we had from this data set was that we could compute the reliability of these measures between the two same-day fMRI sessions. So before we saw it was consistent across subjects. Now we want to see how reliable they are within the same subject. So does your probability of being in the default mode network is the same or is consistent when you do a second scan on the same day? So what we found is that there was a moderate reliability in the data. So we use a measure of intra-class correlation to measure this. You can go into details, but it's basically the within subject, between subject mean squared error. And we found that there was more reliability in the probability of occurrence of state one, the global mode, and the others, probably because we don't have enough temporal resolution to detect them well, the values were moderate. They were not low, but they could be improved. Importantly, if we filter the data, as we show in the paper, this reliability was decreased. So this is the paper I invite you to read, but the LIDA method was first introduced in 2017 in a data set where healthy older adults were compared based on cognitive performance, and we found differences in the occurrence of these states. Then also a paper by Caroline Figueroa where we evaluated patients remitted from depression, and we found that there was a change in this occurrence of these different states. Another paper that we published in Your Image last year used this method and found that psilocybin, which is a psychoactive compound, really decreases the occurrence of the frontoparietal network, a very specific functional network modulation by a single molecule. And this compared with the placebo controls, so a double-blind experiment. Then here is another paper where we applied this to see differences between wakefulness and sleep. Also, more recently, here is not in a psychiatric condition or anything. It's just women that were watching faces of babies, and they had to remember their temperament. And we were able to detect a specific network system that linked the amygdala with the reward center and hippocampus. So it was really interesting to see how these bold phase locking patterns can return meaningful results. So overall, the applications of this method are very broad, let's say. So we can either use it to study psychopathology, so to look for biomarkers of certain psychiatric disorders. We can also use it for therapeutics. They can be helpful because they help us evaluate the effect of a given drug and see if it helps rebalancing the system into back to normal levels. We also have a paper that has just been submitted where we detect interesting and meaningful results when the patients are implanted with deep brain stimulation. And also for overall to understand psychophysiology, so the physiology of the psyche. We also found differences when musicians are improvising jazz inside the scanner, and as in the paper of Jakub Vorisek, we identify subject-specific trajectories. So, as a discussion, and just to finalize, I hope you got interested in this method. So these states of bold phase locking are recurrent, are meaningful, are reliable. And what we find, not only we find these properties, but we find that their dynamic behavior is modulated. It can be changed by task, by psychiatric condition, by mood, psychoactive compounds, visual, auditory, electrical stimulation, and even we detect changes in cognitive performance. So based on these results, and even if you apply different methods to detect, not necessarily LIDA, but different methods to focus on the bold phase locking patterns, I think now the million-dollar question, let's say, is that I'm interested particularly is what is the biophysical mechanism behind this self-organizing pattern formation? So my background is from theoretical and computational neuroscience, and all of this method was developed in order to really understand what is happening in brain activity. So based on the mechanistic hypothesis. And I hope we will soon come to an answer to this, we as a whole community in neuroscience. And so I'd like to thank my network and funding. So I'm at the Life and Health Sciences Research Institute in Portugal, and this is the group with whom I collaborate, especially the group from Oxford, led by Martin Kringelbach, where Jakub is a PhD student, my former supervisor Gustavo Deco, and Bruno Sissak, who helped us, who is a professor in dynamical systems theory and helped us conceptualize this method in the framework of dynamical systems theory. Thank you. That's it.
Hello from Evanston, Illinois. It's a pleasure to be part of this symposium where I'm sure you've been hearing about many of the cutting edge and exciting techniques for measuring brain dynamics. In my presentation today, I'd like to present a bit of a shift from the previous talks and discuss a little bit on how we might compare variations in functional networks across many different timescales. So I'm sure throughout this conference, you've been hearing about how our ability to map functional brain networks with fMRI has really advanced in recent years. So we can now make beautiful pictures of the functional organization of the brain with fMRI, both at the level of the group, as I'm showing you here, and with sufficient data, even at the level of individual subjects. Just to be clear about the methods that I'll be using in my talk today, when we talk about functional connectivity, what we mean is we take a measure of the statistical association in the time course of activity between different brain regions, in our case, just through correlations. So we can measure the bold activity with fMRI from two different regions, like left and right motor cortex over time. We can see how well correlated they are, and we can see that they are indeed quite highly correlated, whereas a region that's part of a different system, like left visual cortex, may show much lower correlations to those two regions. Now, what was somewhat surprising to people who first did this work is that this correlation between left and right motor cortex is present not just when people are moving, but even when they're sitting as still as possible inside the scanner, like a lump. And yet, the spontaneous activity in their left and right motor cortex over time is correlated during this resting state. And these patterns of spontaneous correlations really mimic the patterns of activations that you see during motor tasks. Moreover, we can make these measurements not just for the motor and visual regions I showed you on the previous slide, but many different regions spread throughout the brain, which I can show you here in the form of what's called a correlation matrix, where any given cell in this matrix represents the correlation between a pair of brain regions. Now here I've ordered the regions randomly, but if I reorder them so that I place regions that are part of the same system close to one another, we see this really beautiful structure emerge that we call the block diagonal structure, where we see that there are high correlations along the diagonals, that is within each system, and low correlations in the off diagonals, that is between different systems. Now, because of this nice structure, we can then use a variety of different community detection algorithms to identify systems of the human brain, which I've marked off here with the different red lines and painted on the brain in different colors as shown here. Previous evidence from large group studies, one, for example, here conducted at WashU and one conducted at Harvard, have further emphasized that the functional network measurements that we make can be quite robust. These two different studies identified very similar networks of the human brain on two different populations of subjects collected on different scanners and using two different community detection algorithms. It's perhaps not surprising then that these techniques have generated substantial excitement in the field for our ability to ask different types of questions. For example, how brain networks are affected by brain damage and disease, how they may reflect individual differences in behavior, such as those linked to different traits, how they may change over the course of the lifespan with development and aging, and how they may be modified under different cognitive contexts or with different demands. However, notably, these different types of questions rely on functional networks either being relatively stable over time, so we can make a single measurement of those networks and it can tell us something important about that person at other moments in time, like their disease status or their traits, or they rely on them being quite responsive and malleable to the current cognitive context. And indeed, I'd argue that most papers in the field are at least implicitly motivated by one of these two hypotheses. That is, that functional networks are primarily stationary over time, or that they are reconfiguring substantially with ongoing cognition, mood, arousal, and so forth. And I think these qualities are important to think about because they might affect how we then interpret differences that occur between groups or individuals or conditions. For example, if we find differences in functional networks between a younger adult sample and an older adult sample, is it more likely that these are driven by functional neuroanatomical differences or by what those two samples were thinking about inside the scanner? This may also then change how we think about how functional networks might be best used in personalized medical applications. That is, are they best suited as stable biomarkers or mind reading techniques? Now, I've presented these two extreme examples, but we can actually think about the variability in functional networks as occurring along a continuum, where on one extreme, we might think that certain effects in functional networks are relatively invariant, and that the only differences that we measure are due to measurement noise or sampling variability. On the opposite extreme, we might think that these networks are constantly being modified from moment to moment as we engage in different thought processes and our neurons are firing at different rates. Intermediate to this, these functional networks may change over the course of minutes as we enter into different brain states. And here by brain states, I mean things like states of arousal or task states where we're asked to do different types of tasks. We might think that they change more slowly over the course of the day, for example, with circadian rhythms, that they only change over the course of weeks or years with natural developmental trajectories or with extensive new experience in particular tasks, or that they may be fundamentally stable within an individual, but very meaningfully across individuals. So my talk today will focus on these different aspects of variation. In the first half of the talk, I'll focus on past evidence that there has been for and against stability in functional brain networks, discussing evidence both for changes across task states, as well as evidence that functional connectivity can change during resting state. In the second part of my talk, I'll then contrast the relative magnitude of various of these forms of variation in a single group of subjects measured in a precision data set. Before I get started, I want to make a few qualifiers about the data that I'll talk about today. First off, all of this will be focused on functional MRI data, and in particular based on the blood oxygen level dependent signal of fMRI, which of course, as we all know, is quite slow. And indeed, with functional connectivity, most of the power is present below 0.1 hertz. Thus, these results and the conclusions that we reach may be different from the conclusions that we would reach with other kinds of measurements, like EEG or single unit recordings, which may capture variations at very different timescales of neural processing and have different statistical properties. Moreover, when I'm talking about variations, I'm talking about variations in functional connectivity, not in activation. That is, I'd like to distinguish changes that occur with the second order functional correlation statistic, which is in the brain space squared, versus the first order activation or mean activity level statistic, which is just in brain space. So that brings me to the first part of the talk, where I want to go over some evidence of whether or not functional connectivity changes across task states. So a number of groups have contrasted functional connectivity between task and rest, and it has consistently been found that functional connectivity can vary systematically and significantly between these different task states. These changes occur both within network as well as between different networks. Moreover, individual tasks can also vary meaningfully from one another, not just from rest. And in fact, this likely supports the very accurate decoding of task state that you can have based on functional connectivity data. This can also be seen with variations in very simple task states. For example, eyes open versus eyes closed task states can produce very systematic variations in functional connectivity. So there's substantial evidence for differences in functional connectivity based on task. What about during rest? Certainly our subjective experience is that we are thinking about many different things as we lie inside the scanner during what's called a resting state scan. This work is going to focus on findings from Tim Lauman's study in cerebral cortex a few years ago. So as you've probably already heard, dynamic functional connectivity has perhaps most commonly been studied using a sliding time window technique combined with some form of clustering, like k-means clustering, to identify different changes across different time windows. Now, this data has suggested that there may be additional structure to functional connectivity at rest that may accompany our subjective feeling of changing thoughts during this time window. However, care needs to be taken in the measurement and interpretation of dynamic functional connectivity during the resting state, because unlike task-based modulations, there's no externally imposed perturbation, which means that you have to be cautious about other forms of intrinsic variability and noise that may modify functional connectivity structure as well. This includes things like sampling variability, motion and respiration, as well as arousal that I'll cover in the next few slides. So by sampling variability, what I'm referring to is just the variability you probably learned about in your first statistics class. That is that your sample is just an estimate of a statistic from the full population. And this sample estimate will have a certain variability or error associated with it. And this error goes down with increasing samples from the population. The same is true of functional connectivity when measured over time, which shows a clear progression of decreasing error with additional time points. Since dynamic functional connectivity is usually based on a very small amount of data, we need to be particularly careful about this type of error in our measurements. So how can we identify patterns that appear dynamic but are actually due to sampling variability? Now, one approach is to construct a null model that is stationary, but simulate some noise added to it to see whether or not this simulated stationary null model matches your actual data in different aspects of the dynamics that it shows. Now, this null model needs to be compared to your actual data and needs to be matched to your actual data in a variety of different properties. And I believe Giulia and Raphael will talk in detail about this, but I'll just tell you about one model that Tim Lauman used in his study. Now, this model began with a bold time series that just had random noise added to it. And then these bold time series were constrained to produce the static covariance structure that we see in correlation measures measured over a long period of time. And we're further constrained to have the same power spectral structure that we see in bold activity, that is, with most of the power below 0.1 hertz. Now, we can then compare this stationary simulated data with true data and to see whether or not putative dynamic patterns look different from what we see in this null model. So here I'm just showing you examples of the simulated and real data changing over time. And I'd argue that you might have a hard time picking out which of the two is real and which of the two is simulated from these movies, unless I show you the labels. And in fact, when we apply sliding time window techniques and k-means clustering to these two types of data, we can see that we can identify states in both the real and the simulated data, and in fact that they look quite similar to one another. This suggests that states detected by k-means clustering can be driven by sampling variability. Now, if you're interested in using this particular model, it's available online at this link that I'm showing you here below. What about other more systematic biases in functional connectivity? Now, if you're an fMRI, probably your first thought goes to motion, and in particular, because of its transient nature, motion may look a lot like a dynamic process. When we look more closely, we see that motion does indeed correlate with the kurtosis of functional connectivity, that is the variance of covariance. So suggesting that motion may be related to non-stationarity in functional connectivity data. And indeed, if we remove the periods of motion through frame sensoring, what we find is that kurtosis is greatly diminished. Note that Tim didn't look directly at respiratory-related effects in his data set, but respiration, especially related to deep breaths, may also induce transient patterns in functional connectivity that could inflate dynamic statistics, much like motion. Moreover, of the remaining variation after accounting for motion, Tim also found that much of this might be related to differences in levels of arousal. He looked at this by creating a classifier based on Taliozuki and colleagues' data set, looking at how arousal-related differences are related to changes in functional connectivity data. And he found that sessions which had more light sleep also were the sessions that had higher levels of kurtosis. Now, arousal, of course, may be an interesting neural feature of your data, but it may suggest that an important consideration in interpreting changes in the dynamics of functional connectivity during rest is whether or not they are more likely to be attributable to arousal as opposed to cognitive variation. So here I've shown you evidence that functional connectivity shows significant modulations between task states, but also that functional connectivity within a single resting state can easily be confounded with other factors such as sampling variability, motion, and arousal, making it difficult to find resting state variation linked to cognition. Moreover, I introduced at the beginning some evidence that functional connectivity can also show some degree of invariance across large groups. So the question might be, what are the relative magnitude of these effects in functional brain networks? And can that speak to then the relative value of using functional connectivity to address different types of applications? So that's what I'm going to turn to in the second half of my talk, where I'll discuss some data that we have to speak to this issue based on a precision data set called the Midnight Scan Club dataset that was spearheaded by Nico Dosenbach and Steve Nelson and colleagues at WashU. Now, this data set is particularly well suited to addressing this question because it contains a lot of data from 10 different subjects who each came in for 10 different functional MRI sessions. With this large amount of data, it then becomes possible to create these really high precision maps of individual brain networks. Here I'm showing you 8 of the maps on the outside from 8 individual subjects relative to the group average pattern in the center. In addition to the resting state data that was used to create those maps, these participants also completed 4 other tasks within each session. One was a semantic task in which participants were asked to make a noun-verb judgment on a word. Another was a dot coherence task in which participants were asked to decide whether or not dots were arranged concentrically on a screen. There was a motor task where participants were asked to move their hands, feet, or tongue. And an incidental encoding task where participants were asked to categorize faces, scenes, and words that were repeated over time. Now, this design is nice because it now allows us to look for effects in functional networks that were common across the full group of subjects, sessions, and task states, or selective to individual subjects, sessions, or tasks. For this experiment, we then measured functional networks separately for each subject, for each task, and for each session. I'm just showing you a few examples here, 2 subjects in the 2 different rows, as measured for 333 cortical regions using the Gordon Atlas. Took a few different approaches to looking at the similarity of these functional networks across these different types of conditions. The first was a data-driven approach using multidimensional scaling, as shown here. Now, in these multidimensional scaling plots, each dot represents a correlation matrix, and their position in space represents how similar they are. The closer they are together, the more similar these correlation matrices are to one another. Now, on the left-hand side, I've colored these dots based on which subject they belong to. And on the right-hand side, I've colored the dots based on which task they come from. Otherwise, these 2 plots are identical. And hopefully what you can see at a glance is in these first 2 dimensions, which explain most of the variance in the data, clustering is primarily based on subject identity rather than task. And indeed, we need to go to higher dimensions, substantially higher dimensions, in order to find clustering based on task, with the motor task emerging around dimensions 8 and 9, and the other tasks separating from one another between dimensions 10 and 11. So we can also look at these effects more directly in what I'm going to call a similarity matrix. In this matrix, any given cell just represents the similarity between a pair of networks measured from a particular subject, task, and session. Here, I've ordered them first by subject, within that by task, and within that by session. So just as a cartoon, I want to emphasize that different types of effects would be expected to show different patterns in this map. Effects that are common across the full group of matrices would be affected to increase similarity across the entire similarity matrix. Effects that are specific to a particular individual will increase similarity along the main block of the diagonal. And effects that are specific to particular tasks will be expected to increase similarity along this mini block along the diagonal and the off diagonals relative to that. And of course, we may get interactions between these different types of effects. For example, patterns that are specific to a particular individual in session will show this checkerboard pattern, and to an individual in task will be seen in this single mini block along the diagonal. So what did we see in the actual data? What we found was actually a mixture of these different types of effects. So first off, you might notice that the background is not centered at 0, but is rather around 0.5 or 0.6 correlation, indicating that all the matrices do share a substantial amount of similarity, regardless of which subject, which task, or which day they came from. Now, relative to this group pattern, we also see a big pop-out pattern there along the main diagonal, indicating a big effective individual. That is, a particular subject's networks look most like themselves, regardless of what day or what task they were completing. There's also a small general effective task, so everyone completing the motor task looked a little bit more like other subjects completing the motor task, but it's hard to see on this color scale. Now, relative to this individual effect, we also find a modest interaction between individual and task. You can see this little mini block along the diagonal. That is, a given subject doing the motor task looks most like themselves doing the motor task rather than any other task. There's also a small session level effect for each individual. That is, there was some day-to-day variability, but it's again a little bit hard to see on this color scale. Now, all of these effects were present and significant in this data, but I want to emphasize that they differ substantially in their magnitude, such that functional networks were dominated by stable features like similarity across the group and individual specific factors, with only much more minor or modest effects of day-to-day variability and task state. How are these different sources of variance localized in the brain? We looked at this at a few different levels. First, I'm going to show you how these effects differ based on the type of network. We can separate out networks involved in top-down control, like the frontal parietal, cingulopurcular, ventral, and dorsal attention systems, and compare those networks to networks involved in more basic sensory motor processing, like the visual system, the auditory system, and somatomotor systems. What we end up finding is that there's a similar pattern for both, but that individual specific effects shown here on the right side of the plot tend to be larger in control systems, and cross-subject effects shown on the left-hand side of the plot are stronger in processing systems. We also looked at these effects at different other spatial scales, including at the regional level as well as at the connection level. And again, we found similar patterns. That is, they were all dominated by stable factors with only smaller magnitude effects associated with more transient variation. Also, if we return to the qualification that I made at the start of the talk when I discussed variation in functional connectivity as opposed to activation, what we can also see here is that the patterns of stability are quite different between these two types of methods, with activation patterns showing much larger task and individual by task interactions. So what I've shown you in this study is that functional connectivity networks can have a lot of stability both based on commonalities across the group as well as individual specific features. We also found evidence for modest but significant state level and day-to-day variability, suggesting that there are more dynamic processes that alter functional networks, but that these effects are substantially smaller than these stable factors. Finally, these different effects were localized somewhat differently across the brain, with individual specific features larger in control systems, group features larger in sensory motor processing systems, and task features most prominent in interaction between the two. Now, why are these findings important? Well, I think they sort of have a number of different implications. First off, they suggest that something about the potential neurobiological contributions to functional networks. That is, that they're more likely to be driven by stable, well-conserved factors than sort of ongoing thought processes or cognitive content. This then tells us something about how we might interpret functional network comparisons between groups and states, suggesting that if we do see differences between younger and older adult groups, that they're more likely to be driven by stable neuroanatomical features than what those two groups are thinking about currently in the scanner. And finally, I think this is exciting in terms of the potential to use functional connectivity approaches in different types of medical therapeutic applications, suggesting that their stability, their reliability, and their sensitivity to individual differences may make them very useful targets as biomarkers or for applications such as in neurosurgical planning. So just to summarize what I've shown you so far in this talk, I've given you, in the first half of the talk, I discussed some of the evidence that there has been for and against stability and functional connectivity. For example, showing you that functional connectivity can change across task states, and some cautionary notes about measuring functional connectivity changes during rest. In the second half of the talk, I contrasted the relative magnitude of variations across different time scales, presenting evidence that functional connectivity as a whole is largely stable, showing only relatively minor changes due to more transient factors. So since this is an educational symposium, I thought I'd close with some conclusions and recommendations for then how we might take these findings and think about them in studying dynamics within functional connectivity. The first is probably a point that others have already brought up in this symposium, which is that we should all be careful to define our terminology and our constructs clearly. Oftentimes, it may seem that we're presenting different data and talking at cross purposes, but we're just talking about slightly different constructs. The second is that we might need to be careful, especially during resting state, to address other potential sources of variation that may be present in our data that may not be the variation that we're specifically interested in. This might, of course, include sampling variability, which we might address through various different forms of null models. This could also include more systematic biases induced by things like motion and respiration, which we can address through proper denoising techniques. In particular, I'll point you to these Siric and Parks references here, which have very systematically looked at many different pre-processing and denoising approaches in order to remove variation due to motion, as well as this Power paper here, which has looked at the role of global signal regression in addressing content related to respiration. And finally, that we may also want to address either through measurement or interpretation the contributions of arousal. Ideally, if we would have measurements such as pupillometry or concurrent EEG measures tell us something about arousal levels of the subjects inside the scanner. But at any rate, even if we don't have those measures, it may be important when interpreting the results of dynamic functional connectivity to interpret whether or not findings are more likely to be driven by arousal or ongoing cognitive content. And finally, I'd like to make a plug for shifting in our collection approaches to collecting more substantial amounts of data per subject. In our hands, we've found that this approach has been quite revealing about different forms of variation in functional network data, and in particular has had additional power to detect relatively smaller dynamic differences in the data. For example, we found that task variation is easier to identify with these precision data sets, and indeed, part of this is because a lot of task variation is individually specific. Also close by just putting up here a couple links that may be useful to you, one based on MATLAB and one based on Python, of fairly basic introductory MSC-based tutorials for different ways of looking at your data. Even if these tutorials are more basic than what you would like to look at, I would suggest that you might check them out just in terms of having access to the Midnight Scan Club data and being able to play with it for your own applications. And I'll then just close by thanking my collaborators on these different projects, in particular my former mentor, Sue Peterson, as well as Tim Lauman, who's led a lot of this work on dynamic functional connectivity, as well as my current lab, who's taking on some of the reins of this work to looking at variation in functional connectivity at other timescales. Also close by just putting up my contact information here, so you can feel free to reach out to me separately if you're unable to ask a question at the end of this session. Thank you.